In order to do an RNA-seq experiment, we need to make sure the RNA quality is of is good enough. Because sometimes when you store the sample for a long time, especially if the sample has been made into archival tissues, being able to isolate RNA of high quality is important. Sometimes the RNA can be degraded. This usually happens from the 5' end of the transcript. And so the 3' end is usually a bit more stable. And so after you isolate the RNA from a particular cell or tissue or organ, one good way to check the RNA quality is to run this on kind of a gel. There's some standard machinery to do this. There's a measure called the DV200. This measures the percentage of RNA that is over 200 nucleotide long. And this is just an example. And because most of the genes are longer than 200 base pair, if you have a full mRNA, most likely it will be larger than 200 nucleotide. And so the percentage of RNA longer than 200 is a good measure, and over 30% is recommended. And if you isolate the RNA and you run this, most of the fragments are lower than 200, then you might want to consider whether you will get a good enough readout, because the RNA can be all degraded. In terms of the experimental design, we mentioned in the previous RNA-seq procedure, there are many different designs. Because in the total RNA, the rRNA and tRNA are super abundant. And so there are some experimental procedures or kits that just remove the tRNA and RNA, and you still have the remaining total RNA, including all the introns. So some experimental effort might be interested in looking at those intronic RNAs. The most popular or widely available data on RNA-seq is poly-A RNA. This basically is using the poly-A to select the mature mRNA after splicing. This really enriched for exons, and there are no tRNA or RNA, and most of the introns are also removed. If you are interested in understanding strand, the specific strand that creates this RNA, you can also use strand-specific or directional RNA sequencing. This is most useful for identifying novel long non-coding RNAs, which may or may not have a standard coding frame, which give you the peptide sequence based on the RNA, because the link RNA may not be translated into a protein. This will be additional experimental step. So depending on the different need, you might pick a different experimental technique. Over the years, this RNA sequencing cost has decreased dramatically. In 2008, when RNA-seq first became available, the cost could be as high as $2,000, and it continued to decrease. And nowadays, there might be sequencing companies. You send them your RNA sample, they will make the library and do the sequencing for you for less than $200. It could be as low as like $150. But then you have to ask, what does a $200 RNA-seq sample really mean? Because there are many different ways of doing the sequencing. First, you have to decide whether you want to sequence single end or paired end. Remember, for the fragments that you get before you put on the sequencing, there are sequencing adapters from both ends. Depending on the facilities you're sequencing, you could just sequence from one end, but you could also sequence paired end. Usually the second end quality is slightly lower than the first, but overall you will get twice as much data. In terms of sequencing depths, this is also a significant difference, because nowadays a lot of people can multiplex many samples to be sequenced on the same sequencing lane. And so depends on whether they put 2 samples in a lane or 20 samples in a lane, you might get a different number of reads per on your own sample. And so when people tell you this is $200 per sample, you have to ask, do I get 20 million fragments or 50 million fragments, right? So For bigger projects, people might want to sequence about, I think that most of the popular ones are at 50 million. You will get good, very solid differential expression. If you want to get just kind of a general, very quick idea of your expression, 10, 20 million fragments are also good enough. But if you really want to look at novel RNA or splicing or de novo transcript assembly, you might want to sequence deeper, say 100 or 200 million fragments. A third important factor is sequencing length. In the early days of high throughput sequencing, we only had the machine only allow you to sequence 30 base pair at reliably. Later on, it's changed, extended to 50 base pair, 70 base pair, 100, and now many standard sequencing machine can give you 150 base pair fragments. And so you can imagine if at $200, I can get 500 million paired end 150 base pair fragment, you really get a lot of data. Whereas if they only give you a single end 10 million at 30 base pair, then you don't really have a lot of data to really analyze. So before you just get the price, understand what you are really getting from that sequencing reaction. In terms of experimental design, for RNA-seq, we really recommend having replicates. So the need to have replicate initially came from expression microarray days. And at that time, people also do technical replicates, which means after you isolate the RNA from a particular sample in the early microarray days, because the gene chip, depending on which graduate student spotted the array, if you try it on two different arrays, you might get very different results. And so at that time, people do have different technical replicates from the same RNA sample. But with RNA-seq, this is rarely done, which is the same RNA, but different library preparation and sequence separately. But we don't really recommend this in a real RNA-seq experiment. Most of the time for RNA-seq, people recommend having biological replicate. And there are also different levels of that. The first level is if you are dealing with a cell line, you grow the cell in different petri dish on different days, and you isolate the RNA in different batches on different days. And usually the treatment and control are still done in the same cell grown at the same day using this identical reagent, but you might have replicates that are done on different days. That's kind of the very basic things, because you are really assuming the cell lines are very, very different, and hopefully you are using the same reagent, same cell culture conditions. With other situations, such as in animal cases, these are slightly better biological replicates. For example, when you have lab animals, they are genetically identical, and you try to buy the mice from some company, and they have the same age and same lifestyle, and so they are fairly similar, but maybe they still exercise differently. They hang out with different friends or something. So there are slight differences, but these are better biological replicates than cell culture. And the highest level of biological replicates are, different human individuals, say tumor samples from different patients. Patient might have different racial, gender, many other different lifestyle, geographical locations, different hospitals. And so these actually have much bigger variabilities. And so depending on the level of replicates, this is what we would recommend. If you don't do replicates, just to look at, say, my drug treatment and control, two conditions, one replicate, this is only good for exploratory assays. It's not really good for publication. For example, you might want to see, do I want to treat myself for 3 hours or 6 hours or 24 hours? You can just generate one replicate and just roughly see the difference. And then later on you decide, okay, I'm gonna use, say, 12 hours for all my later experiment, then you can generate biological replicates. If you are looking at cell line samples, 2 replicates probably is good enough, at least is publication ready if you are able to do the experiment fairly consistently. When you have animals, usually we would recommend more than 3. In fact, in those situations, you might want to have 10 mice in each arm, or maybe 9 mice in each arm, and then pool the 3 mice together to form one group to do RNA-seq, another 3 in one RNA-seq. So if you have 9 mice, you can probably generate groups of 3, each with 3 mice, and you have 3 RNA-seq result. With human, because human variations are much, much bigger, you might need a lot more samples in order to see a robust biological difference between the samples. But replicates are definitely needed